So now we will move to the panel discussion. And Jim, it's possible that at the Q&A at the very end of the panel discussion, there may be some more device-specific questions. So please stay online.
Okay, of course, Henry. Yep.
So today we're very happy to have a very comprehensive set of experts in our panel. So first of all, we have all of our principal investigators from all 3 study sites, Per Svenningsson, Håkan Widner, and Philip Sheperians. And maybe the panelists at this point, you could start turning on your webcams and microphones. We don't have Juha Rinne online yet. Juha is online? No?
No.
So hopefully he'll be joining us in a few minutes. So Juha is a leading expert in PET imaging, and we were hoping to have him involved. He was still in contact with us just shortly, so maybe something unexpected has come up. Then we have Göran Lind, a neurosurgeon who was involved in this study at the Stockholm site. And we have Craig Cook, the CEO of Herantis, and Paul Skinner from Renishaw Neurosolutions. And we're very happy to have Alan Wone from Bristol, who was the principal investigator in the GDNF phase 2 study published recently. So,
Hello, it's Juha here. Can you hear me?
Now we have Juha online. Juha, you just made it. I was just wondering what we have to do without you.
No, I have some problems, getting the video on.
Okay, that's fine. We can hear you, so that's good enough. And if the video doesn't work, we can live without it.
Yeah. I think that you don't miss anything not seeing me.
Okay, thank you. So maybe before we go to the questions, I would like to ask all the panelists to briefly, just 1 or 2 sentences, to introduce yourself so that the audience will learn to know you, your role perhaps in this project, and so on. So maybe we can start from Per and then go Håkan, Philip, Juha, and so on.
Yes, I'm professor of neurology and follow mainly patients with Parkinson's disease at Karolinska University Hospital in Stockholm.
And Håkan Widner in Lund. I'm also a neurologist, movement disorder specialist. I've been involved in regeneration trials for Parkinson's over the years.
I'm Philip Sheperians, adjunct professor of neurology in Helsinki University Hospital. I've been the PI in Helsinki, and I'm working on gut microbiota and Parkinson's, so my clinical focus is definitely also in Parkinson's disease.
Yeah, I'm Juha Rinne. Sorry about joining a bit late. I'm a neurologist working at Turku. I'm working at the Turku PET Center, which is a research center for imaging. And I'm a neurologist by training, and my role in this project has been coordinating the PET imaging of Finnish participants.
I'm Craig Cook. I'm the CEO of Herantis. I joined the company roughly 4 months ago and delighted to be part of this webinar. Thank you.
Yes, and I'm Göran Lind. I'm a neurosurgeon at the Karolinska University Hospital and have performed the implants here.
Yeah, good afternoon. I'm Paul Skinner. I'm General Manager at Renishaw Neurosolutions and been very pleased to be involved in this study from a device perspective.
And I'm Alan Wone. I'm a neurologist in movement disorders in Bristol. I run a large clinical service, but also do research, including being responsible overall for the GDNF study, which was published shortly before this study concluded on CDNF. And I very much enjoyed interacting with Per and Håkan and other members of the Herantis team over the years whilst you've been doing your studies. And as someone who wasn't involved directly in this study, I just want to say congratulations to you on reaching the endpoint and at meeting the safety primary outcome.
Very good. Thank you, gentlemen. Then we will move on. And as the first question, because we have quite diverse backgrounds in the panel, I would like to ask for your personal viewpoints to the TreatER clinical data. What are the important points in this clinical study that you would like to highlight? And maybe since Per was already presenting, maybe we can start from Håkan and Philip, and then go Per, Juha, Göran, and the rest of the gentlemen. So Håkan, please.
Yeah. Well, I think one could say that there are diverse responses. I believe that we have had a couple of clear responders in the study, and they have been doing very well. At the same time, we have had patients that have not responded, even though they have got an active drug, and we need to find out why. So I think there are biological principles here that are very interesting and important, but we need to understand why not all are responding in the same way.
Philip.
Yeah, maybe I could. I mean, I agree with Håkan. For me, I think the most important outcome of the study is obviously that there were no CDNF-related adverse events that would be, you know, so strong that they would, you know, prevent this from being further developed. So I think the safety aspect is obviously most important. Yeah, otherwise, I mean, the data, the outcome data in terms of the symptoms, it's heterogeneous. It has been a small study. I mean, it has been encouraging findings in a subset of subjects. I mean, it's too early, I guess, to say anything in general about the efficacy here, but we can definitely say that we have no indication of tolerability problems related to CDNF. Obviously, the way of application is it's not the easiest, so it has been quite demanding to conduct this study, and it has been associated with certain risks as well. But I think the risk management has worked fine throughout the study, and we have all learned a lot. And so I think there is good hope to see this going forward.
Yes, first of all, I would like to say that this has been a demanding study that has needed collaboration with different sites, and now we have shown that it is feasible to perform this kind of study and can give interesting information, and I think the results are encouraging, and I think first of all, the primary endpoint, the safety, was met, but not reiterating the other speakers, but I think that we really need to understand more clearly why the response, both in clinical and PET respect, has been very variable between the participants.
Would you like to add something?
Yeah, I can just echo what has been said. That I mean, it's of course encouraging that there were no adverse events directly related to CDNF, and it feels safe to work with the substance. And I also think that, I mean, the support from you and also Renishaw has been very good, because it's, as said, it has been a very demanding trial, and also we have had many visits for the patients and so on. But I think it has run nicely. And then I again emphasize what has already been said, that we need to identify optimal patients for this therapy in the future, so we can really stratify the trial. So, and it will be interesting now to see all the final analysis that you're performing, and see if we can get any more clues for future trials.
Göran, what about the neurosurgery perspective?
Yes, that's was quite interesting to hear Jim Baker tell about the improvements that are coming up. This is more demanding surgery than the everyday DBS surgery that we perform on many Parkinson's disease patients. So it's good to improve the surgical process here. However, I would like to point out that it worked out nicely with surgery, but both implanting and explanting has worked out nicely for all the patients. So this could very well be an interesting development in the future.
Craig, would you like to go next?
Yeah, so I think most importantly, clearly safety was essential for us to establish. It's always an anxious time when a company takes a drug from preclinical science phases into human beings for the first time. So that's clearly a key output for us. Second of all, I think the understanding of the need to target patients earlier in their disease was again a key theme that is going to be an important learning for the program. The dose response information was similarly interesting in that it tallied with the preclinical findings, so was not unexpected from that perspective. And I think the final factor, the final element to add to that was it clearly wasn't an efficacy study, but nevertheless it was encouraging to see that there was no worsening of the disease during the duration of the study in a disease that typically worsens. So I think that those would be my 4 key takeaways from the program.
Paul, what about Renshaw?
So yeah, I think we were very pleased with the outcome of the study. Obviously, throughout the study, there were things that we learned, and I think the opportunity to introduce some of those changes as we went through the study was really important. And so, as Jim had mentioned, things like introducing the port drape to improve the method of attaching the giving set, and to also be able to do some retraining of the clinical staff with new cleaning methods was really important. And I think then that after we'd done those, to see the reduction of adverse events that we had after that was really important for us. So there's definitely lots of learning that we've taken from this. And also I think the collaborative nature that was really important to go through this, and to be able to do this study across 3 sites was very important to us as well.
Alan, would you have any comment for this question briefly?
I think much has been said, but again, congratulations to the whole team of investigators on delivering what's a very tough study and a first-in-human study for CDNF. I mean, I think it sounds as if from what was presented that Renishaw and the neuroinfused system has made progressive steps forward during the course of the investigation. I guess contrasting with the GDNF study may not be valid for some of the reasons that were different in the biology that was outlined for us by Mart at the beginning. I think I'd be interested to know what Juha makes of the PET data. That possibly seems less consistent than you might have expected. I'd be interested to know what the test-retest is of that imaging using the ligand you use. So I think congratulations on delivering the study, and it sounds as if there have been some progresses made with certainly progress of the safety of CDNF when first used in humans and the delivery device system. I think I agree the sample is very small for efficacy data. I wonder how you might take this forward now based on the efficacy that you have and the PET imaging that you have, how you are going to stratify. I don't have anything else.
Thank you, Alan. So let's move on to the next question, which is how does this clinical data affect the sponsors' future plans? And this is pretty clearly directed to Craig and Paul. So would you like to take this question?
Yes. So as I indicated earlier, we were very pleased with the outcomes of the study and having established safety in the study. And a massive thank you to the patients that participated in such a challenging and intense study, to the medical teams, to Rainy Shaw. It's a major achievement from our perspective. So we will forever be grateful for the collaboration and the participation of patients and all the stakeholders. So from our perspective, it's a solid foundation. We believe that we now have the elements to build a successful program moving forward. And the focus for Herantis will be we're going to move to alternative administration routes. So we'll be moving away from the need for surgery and focusing on intranasal administration and subcutaneous administration. We have solid scientific merit and rationale for those decisions. We have data on those administration routes with CDNF, and we also have the necessary intellectual property and patents filed for those administration routes. And essentially, we're looking to the key objective is to try and expand the target population to earlier disease patients. And that's the key driver for our decision to pursue these alternative administration routes or expand to these alternative administrations. So effectively, we're backing CDNF as a standalone product moving forward without the need for surgery.
Paul.
Yeah, so moving forward from this study, there's obviously learning that we've taken out of it, which we're incorporating back into the device. We've gained data from this study that we will be able to use to move forward with approval of the device. But the device on its own is, doesn't give the clinical benefit to patients. So we will continue to work with pharma companies for using the device as they progress different drugs through that need to be delivered directly into the brain. And I know that from what Craig has said that, CDNF is looking at other routes forward, but there are also a number of other drugs that are being developed that need to be delivered directly into the brain, whether that's for Parkinson's or for other conditions. And we'll continue to work with pharma companies. And really the data that we've got from this is really helpful for us in progressing it, both from a development perspective and also from an approval perspective. And I guess I'd also like to just reiterate that thanks to the patients and their families and to all the teams that have been involved, really, because I know this has been demanding studies on all those fronts. And from a Renishaw perspective, we'd just like to thank all of those involved in all they've given to make this a successful trial.
Very good, thank you. So next one is a specific question for Alan. You have a excellent visibility to the previous GDNS study as the principal investigator. So how, from your perspective, how does the trial and the data, the TreatER trial, compare to the previous GDNF phase 2 study, and what are the important differences and similarities between these two studies? And also related, there is one question from the audience. I put it here in this part because it relates to the placebo effect in both studies. So would you like to comment that as well?
So the similarity for the GDNF and CDNF study, as I say at this point, without having seen all the data, is that the trophic factors themselves were well tolerated. So that's important. The Another similarity seems to be, if I've understood correctly from the presentation, the last presentation that we had before the panel discussion, is that the infusions themselves, if you had decided to go ahead with an infusion approach, were well tolerated. I think in both studies, there were problems with the port sites. It sounds as if, from what the Renishaw team presented, that there are some approaches to getting that better. Certainly an important and necessary to explore serious adverse event with the cerebral, intracerebral abscesses that were seen in the CDNF study that we didn't see in the GDNF study, and there may be reasons for that. Perhaps needs to be reviewed. And then in terms of the efficacy outcome, it's difficult to compare to some extent the two groups, because although the GDNF study was still small, it was somewhat bigger. So we had 20 subjects in each arm. We didn't have a different dose group. So we only had a single dose group in GDNF versus the 2 dose groups here. Like the CDNF study, the GDNF study showed that some people appeared to respond well, although we can't prove, I don't think in either study, that that is a definite treatment effect and not just a very large practice effect or placebo effect. But there appeared to be some people who responded. As yet, from the data I've heard for CDNF and for GDNF, we can't identify why the people who respond better. And that is a problem, because you don't know how then to enrich future clinical trials. One potential, I think it might be reasonable to say that the magnitude of benefit on average in both of the studies was problematic at this stage in terms of not reaching statistical significance in either study, although I appreciate the CDNF study was not powered for that. And in terms of magnitude of benefit, obviously we're quite some way away at the moment from the magnitude of benefit that you're getting with the proven device-assisted therapies like DBS or duodopa intestinal gel. So that's an issue for future studies with people with Parkinson's coming forward, in a way that over the time course of the study, they're unlikely to do as well as they would do if they went for a conventional device-assisted therapy, but of course those aren't neuroprotective. So there is something about the size of the effect at the moment appears to not be consistently big enough anyway, neither to reach significance nor to be a contender with device-assisted therapies. And then there's the interesting thing about the PET data. So something to know that's different between the studies is that different radioligands were used. So in the GDNF study, we used something called fluorodopa, and we did see a very robust and significant signal that was statistically significant and meaningful in the size of improvement in the group. And with the DAT tracer in the CDNF study, I think it would be reasonable to say you don't currently have that, but Juha is a much greater PET expert than me and can comment, but you don't consistently seem to have that. So at the moment for the GDNF study, I think we can at least say we've got target tissue engagement. With fluorodopa, you may be binding to serotonin nerve endings, is what you're showing. So that may explain the difference. I think it probably suggests if we did another GDNF study, we should use that to check that our fluorodopa finding was actually showing us something happening to dopamine nerves. But I think in general that the safety is encouraging. The magnitude of benefit is problematic at the moment in both GDNF and CDNF groups, and possibly for CDNF at the moment. I think the DAT imaging perhaps raises some concerns. I don't know whether that's felt to partly be due to distribution as well, although the infusion approach was the same for both, because of the way GDNF binds to heparin binding sites, whether that whereas CDNF does not, whether that altered the DAT change, whether the infusate was actually going over a much broader distribution. I mean, one of the things that's interesting is that a couple of reasons that GDNF was suggested maybe didn't work was because with the GDNF study we used a trophic factor that had been made in E. coli rather than mammalian cell lines, and also we may have underdosed, and in the CDNF study you've used higher doses and you've used mammalian cell line, and yet still you're not seeing perhaps the magnitude of benefit that was hoped for, albeit that this is exploratory. So I think there are things to learn from these studies, and those are the similarities and differences as I see them at the moment. I'd be interested to hear what others think.
Maybe Juha, you could comment a little bit on the PET tracer question here.
Yes, well, thank you. Yes, perhaps not going into too much into detail, but like Alan was pointing out that fluorodopa is not specific for dopaminergic system. It in a way tells about the activity of a certain enzyme that converts aromatic amino acids, and therefore it can also reflect the action of serotonin and noradrenaline neurons, as well, of course, as dopamine neurons. In the striatum, most of the terminals are dopaminergic, so the bulk of the signal is from dopaminergic neurons, but it can still stem out from other amino acid transmitters as well. And then on the other hand, we know that which binds to the presynaptic dopaminergic terminals, that there might be some compensatory changes in these binding of these ligands during the Parkinson's disease, because in a way, fluorodopa is telling about the synthesis of dopamine from levodopa, and that might, at least according to animal studies, that might be upregulated in Parkinson's disease. Whereas on the other hand, because that is partly involved in taking the dopamine back from the synaptic cleft, so therefore, it might be reasonable in Parkinson's disease to downregulate that binding, and therefore, it sometimes makes it difficult to interpret the results of these different ligands, and perhaps one should, of course, in a optimal world, one would do like a multi-tracer study, or then perhaps to use a VMAT tracer, but in any case, one thing which I think is good to remember that these subjects in the CDNF study, they actually had a very advanced degeneration of the striatal dopaminergic system. So it was quite surprising that even in such participants in the mid-dose study, there was a clear increase in the average DAT binding, which might tell us something about the state of the presynaptic terminals.
Thank you. That's very, very interesting, and of course very important. PET is an important endpoint for these type of studies. So in the interest of time, I will actually skip this question because I think we already partially covered some of the key learnings for different consortium members. I would like to ask all of the panelists now, how do you see the future of neurorestorative disease-modifying therapies in Parkinson's? Stepping a little bit outside of CDNF, GDNF, and so on. So what would you see are the important next steps in the field? Maybe we can actually start from Pat now.
Yes, no, but I think this is, the really, important task for us, being seeing patients and seeing how they progress to really find therapies that at least slow down the disease progress, and we need to continue to do this type of trials that we're doing. Like we have said today, I think it's more reasonable to believe that the therapies are successful if we start earlier on in the disease course. And but I think this is, of course, I mean, something that we need to pursue. We need to, at some stage, perhaps also thinking of combining therapies, because it may be that this idea that one single molecule will be sufficient will not turn out to be. The disease has so many different adaptations in different cellular systems, so we need to perhaps start to think at some point of combining some of these restorative therapies. Although I think from a rational standpoint and the clinical disease design, we need to continue to also, of course, study them individually. So I think this is, I mean, we just need to be stubborn, we need to be patient. And that's something that I think both these trials have been good, both the GDNF and CDNF. We have, after all, had some patients. We continue now with the extension study to continue to follow our patients and so on, because we know that the duration of time that it takes to progress in Parkinson's disease is so long. So that's another aspect that with that. But where I think this trial has been very good, and also the GDNF. I don't think we waited for enough time to at least see some signal. But again, I will on my side, we just need to really, I mean, stay focused and continue to do this work. And again, like we do now, collaborate in order to find, I mean, it's very difficult for one site. I mean, now Alan was extremely impressive in Bristol when he could run a trial by himself, but I think these collaborative efforts are very, very important also for the field.
And maybe here I'll step in a little bit and also address an audience question, which is how long will the patients continue to receive CDNF in order to collect long-term effects? And maybe to correct a slight misunderstanding here, the follow-up study does not include any further dosings. So actually the patients have stopped receiving CDNF, and it's just follow-up how their condition develops. And maybe we can now continue to Håkan about the important next steps in the field.
Yeah, I agree very much with what Per Svenningsson said. Restorative and disease-modifying therapies are really important. We have actually very good treatment options at the moment with all the medications and deep brain stimulation and other devices. But it's the progression of the disease that is, if we could modify this and essentially stop the progression, that would be very beneficial, and then we could use all the tools and medications that we have. The nature of the disease is very complex, and it's really asking a lot that one single approach or one single medication could affect and stop the disease. Just making an analogy with oncology and cancer treatments, it's very often a combination of 3 and 4 different approaches, or in organ transplantations, you usually use 2 or 3 or 4 immunosuppressive drugs to really control the complex responses. And that is extremely difficult to tease out which approaches one needs to combine to get an effect. And it's very hard to do it in the clinic. And it's also a little bit, it's usually different companies as well. So it not only sort of a complex biology of it, it is really complex and maybe only academic or European Union type of studies that actually can perform studies like that. But very interesting.
Yes, I would like to continue from where Håkan stopped. I think the comparison to the cancer field is very important because I mean, major advancement in cancer treatment have been made when biomarkers were found that could identify patients with certain cancer subtypes that are sensitive to certain treatments. And so I think in Parkinson's, we're currently facing the problem that we are subsumming a lot of different patients with probably different underlying pathologies and different pathways to Parkinson's, and we all call them Parkinson's disease, even though the underlying problem may be different in different patients, the cause for the disease and also the factors that determine progression may be different. So I think in terms of disease modification, as everyone else also said, I think we need to go earlier, so we will have a probably better chance of picking it up. We need to follow up and treat the patients for a longer period of time. And I don't think there's many other options until we find reliable biomarkers or other indicators that allow us to enrich cohorts with patients that likely progress quickly. If we get those, then it will be helping us, you know, to have smaller and shorter trials, I guess, and we will get answers hopefully more quicker. But currently, I don't see that we have these tools really available, and this will be a major breakthrough if we end up having such tools. Yeah, otherwise I just think it's going earlier and treating longer and following up longer. I'm afraid that's what I see is the current choice until we have such stratification biomarkers.
Juha, Göran, Craig, Paul, would you like to add something?
Well, you know, I can just first I can say that I can echo what Philip was saying, because you know we know that Parkinson's disease is a long-standing progressive disease, and there are individual differences in the speed and trajectory of the progression of the disease. So I think we clearly need studies with larger number of participants to get out reliable picture on the effect of these kind of therapies. And we need more longer studies, even though this has been like 1 year study, but 1 year in the course of Parkinson's disease is still relatively short, and knowing the individual differences in the progression rate. So I think we need longer studies, and of course starting earlier. And then of course the other thing is that we need to try to understand what might be the biological reason behind this variable response that has been seen in these different disease modifying trials, both in clinical and imaging outcomes.
Very good. Actually, in the interest of time, I think we got a pretty good idea on what's important going forward. And then we have a few minutes left, and I would like to leave some time for questions from the audience. And let's start with the device-related questions. So first of all, there's a question, and I think it might be actually nice if Göran, you could take this one. So what happens to the device implanted in the patient's brain after the CDNF trial? Does it get removed or left in?
It has been up to the individual patient to decide, and at least in Stockholm, all the patients we've had have decided to have them removed. And this has been done completely without any adverse events. So it has worked out nicely, removing the implants as well.
Paul, is the device in clinical use for brain cancer? That's another audience question.
Yeah. So we've done some use of the device for treating brain cancer. On humanitarian grounds so far. So we've had about 30 patients that have been treated using the device for brain cancer. Yeah. And just going back to the question of removing the device, so there were some patients that had just the port removed. So one of the criteria for approving the study was that the port had to be removed at the end of it, but some patients in the other centres have had the catheters remain in place, and the actual catheters are made of the same material as another product that we've got, which is a long-term implantable, which is used with implanting of DBS electrodes.
Thank you. Then maybe, Per, you could take this one. Did anyone experience any symptoms such as Lermit's sign, as was experienced by some of the GDNF study participants?
Not at our site, at least. I don't. Philip and Håkan should also comment, but no.
Not that I'm aware of.
No, not in Helsinki.
One of the first speakers today mentioned that neurotrophic factors do not work when more than 80% of dopamine fibers are lost. Does this explain the limited efficacy of CDNF in the present study, and also the lack of efficacy in GDNF study? Does animal studies support this? I would first like to mention that yes, we believe that if the loss is advanced, there's really not much dopaminergic innervation left or fibers left. It's expected that the efficacy of any growth factor treatment would be limited. Now, any of the panelists, would you like to add on top of this, and maybe continue a little bit on how it would be best to define a study population for next studies with CDNF or GDNF in terms of the stage of disease? Who would like to start?
I mean, I think one of the things that's helpful, Henry, is in addition to all the things that have been said that you could do in a future study, the other thing is bigger numbers. So these are obviously very small number studies when they're surgical studies. And one advantage of adapting the delivery approach that as you're proposing to do is that hopefully you'll be able to do much bigger studies, larger numbers of subjects. And perhaps another thing to say that's common to the GDNF and CDNF study is, of course, as for other neurorestorative studies, we've all been using the same outcome measure. And without wishing to criticize the UPDRS, I think there probably are problems with that and the noise inherent with that for the part 3 for doing these type of investigations. And so that, in addition to stratifying studies and larger studies and longer studies, and earlier patients, possibly different outcome measures may be important. And obviously you've made a first start here. I think this is probably the first neurorestorative study to use a wearable that you've just reported today. But I think that's going to be important as well for the future.
Yeah, it's Juhari here. I think that one of all the clinicians that are treating patients with Parkinson's disease really see that there is a huge individual variation in the progression of the disease. And like Philip was stating earlier, that I think that if there are some hints or indicators that some individuals might progress more rapidly, then you might try to target these individuals to be able to get the effect and earlier and faster, because I think that otherwise you would need quite a large number of participants because the variation in the natural progression is relatively large.
Maybe I could shortly add to that. I just mentioned that there is no biomarkers to predict progression. One thing that one could consider is Parkinson's patients that have REM sleep behavior disorder. Those patients tend to progress quicker than Parkinson's patients that do not have REM sleep behavior disorder. So that has been considered as one marker that could indicate a higher risk of progression. So it could also be used for stratification of such a population.
Yeah, one general comment on restorative approaches is that I think one should try to administer it as soon as possible, but then that precludes sort of very invasive methods. It's very hard to justify major surgery with potential risk of very severe complications in a relatively mild condition of Parkinson's. that if we had a perfect drug, one might change that because in, for example, multiple sclerosis, if we have a severe bout of multiple sclerosis early on, we are not hesitating on advocating very severe but very effective treatments, because we could alter the disease completely by bone marrow transplantation or advanced monoclonal antibodies. But we're not there yet in Parkinson's, I would say.
Very good. So it looks we're close to 6 p.m. finish time. So maybe a couple of quick questions I will try to answer in the very end here. So there's one question: did the team identify any common factors amongst those who had positive outcomes other than time after diagnosis? Any common factors amongst those who showed no real change from expected? This is something that we're currently working on. We still don't have all data in. There's some for example, the CSF proteomics data is coming. We're waiting some of additional alpha-synuclein data to come in, and we certainly will be looking into this when preparing the manuscripts after New Year. So far, we haven't found anything obvious, and I believe this was also the case with the GDNF study, that there was no common denominators found for those the responder population of the patients. And then finally, there's a question: technical problems prevented some people from joining the first half of the webinar. How will slides be made available to viewers? We did mention this after the first break. We will make a record of this webinar and the slides publicly available. Please allow us a day or two to have all the materials, but it should be found on the TreatER website and possibly links from Herantis and Renishaw website as well. So this brings us to the end of our 3-hour program. I would like to take this opportunity to thank everyone in the audience, the panelists, the presenters, and on behalf of Herantis Pharma and the other TreatER consortium members, I would like to say special thanks once more to the patients and their families who participated in this clinical study. This was a very demanding study from the patient perspective and also from the staff perspective at the study centers. And without the courage and perseverance of the patients, this study would not have been possible. So once again, thank you. Also, the staff at the study centers and other partners and consortium members have been critical in successful completion of this study. Many thanks to everyone for smooth and professional collaboration. It has certainly been a pleasure, and I hope we are looking forward to working with all of you in the future as well. Stay safe and healthy in these unusual times. Thank you and good night.
Good night.